In this video, we'll talk about DNA transcription. And DNA transcription is a process, right? So transcription is the rewriting of something. So transcription of DNA is just kind of rewriting some of the information in DNA. That's all it is. But let's first talk about kind of why DNA is important. And I'm sure that you've heard that your DNA is kind of what makes you who you are, and it's kind of true, kind of not. DNA in the order of bases that encode your specific DNA, so the C's, G's, A's, and T's, the specific order of those bases holds the information to create proteins. And again, that specific order creates very specific proteins. And it's really your proteins that make you who you are. But again, the kind of identity of those proteins is held in the information in your DNA. So DNA is really important. And now if we think about getting from the information of DNA to making your actual proteins, transcription is just one of the steps in between. And so let's talk about the differences between DNA and RNA, because that will become important. So RNA is more or less the same as DNA. So here's RNA up here, here's DNA over here. There's only three main differences. One, DNA, the D in DNA stands for deoxyribonucleic acid, and that's because it uses the sugar deoxyribose, this guy down here. Well, R and RNA just stands for ribose, ribonucleic acid. So the sugar it uses is ribose instead of deoxyribose. And the only difference between ribose and deoxyribose is that on the second carbon here of ribose, there's an OH, where there is no OH on deoxyribose. That is literally it. So, difference number one between DNA and RNA, DNA uses deoxyribose, which does not have an OH group on its second carbon, and RNA uses ribose, which does have an OH group on its second carbon. Difference number two is that RNA is single-stranded, right? It does not form hydrogen bonds to make a double-stranded RNA molecule with kind of like the complementary strand. Stays single-stranded. The third difference is that in RNA, the base thymine is replaced by the base uracil, and you'll see over here that they're very similar in structure. And so it is the uracil in RNA that can form a complementary bonds with an adenine in DNA. Okie dokie. So let's talk about transcription itself. So in transcription, there's a protein, an enzyme called RNA polymerase, and it attaches to specific regions of DNA. And what it ends up doing is making an RNA copy of your DNA. And that RNA is called messenger RNA, or mRNA. So when the RNA polymerase attaches, it kind of unwinds some of your DNA. And then, similar to DNA replication, it inserts complementary ribonucleotides. So instead of deoxyribonucleotides, which are the nucleotides for DNA, these are ribonucleotides, the nucleotides for RNA. And RNA polymerase inserts the kind of complementary ribonucleotides in with the DNA. So if there's an A on the DNA, it inserts a U. If there's a T, an A. If a C, a G, right? And in that way, it's forming these complementary bonds as it goes, and it's connecting those ribonucleotides together to make a strand of mRNA. And one nuance, again, is that RNA polymerase can only create RNA going from 5' to 3'. Right? So in this picture, it's kind of left to right. But remember that for this complementary bonding to work, the strands have to be anti-parallel. So if the RNA is being made from 5' to 3', it means that the DNA is going from 3' to 5'. And so RNA polymerase always reads DNA from the 3' to 5' so that it can create RNA from the 5' to 3'. And it can only do this kind of with one strand of DNA, and that strand is called the template strand. And the other strand, kind of the one that the RNA polymerase is not reading, is called the coding or lagging strand.